Hello young guys, you are welcome in the biology classes organized by Society of Career Technology and Jan Vikas Mahasabha. I am the resource person, Dr.Agam Dayal. We have discussed out of 4 parts of reproductive system reproduction, first part showing asexual reproduction. Today we will discuss about second part about sexual reproduction. This lecture is for class 10th of CBSE students. One very interesting thing in CBSE book is that they have described 4 hierarchies. In one hierarchy, they have discussed about DNA replication, and then in second stage they have discussed about involvement of nucleus during fertilization process, reproduction process. Then they have discussed about cytoplasmic changes in the gamete. Then they have discussed the reproductive organs this way. Now, one very interesting fact is that in asexual reproduction, only one parent is there which produces one of its cell from its body. It develops into a new organism. So this new organism is a clone of its parent. It is genetically similar. But for natural selection and evolution to occur, variation is essential. This system is not providing opportunity for variation. So nature selected another system in which 2 individuals, male and female, produced a gamete which contained only half of the genetic material, and then they combine with each other to form a zygote. Here also a variation is there. In some of the lower animals, we have external fertilization in which both male gamete and female gametes are released outside in aquatic condition in water, and there they fuse with each other to form zygote. For it is external fertilization. But in this system, there is a risk for developing embryo that it can dry, it can be wet, it can be fed by predator or parasites. So it was risky. So nature developed another system in which the female individual released female gamete, but it was retained in the body of the female itself. Father produced male gamete which was released and then passed to the oviduct of the female. There both of them united to form a zygote, and then there were 2 possibilities. Either it develops into a new organism that is born. This condition is called viviparous. At this level we have 3 systems in nature. One is oviparous, in which the both egg and the sperm are released outside the body. There is external fertilization, or sometimes there is internal fertilization in which the male gamete travels up to middle part of the oviduct, but the egg is released outside in external environment. There is external fertilization. So when egg is released outside, it is called oviparous condition. There is a condition in which there is internal fertilization. Ovum is released into the middle part of the oviduct. The sperm travels here into the middle part of oviduct, fertilizes it and forms a zygote. Development takes place by the reserve food material yolk present in the egg, but there is no placenta. Here also, after some time, a new young one is born. This is found in certain fishes and reptiles called ovoviviparous. Then another condition is viviparous, in which there is internal fertilization and there is placental connection, so the uterine wall of the mother also provides nourishment to the developing embryo, and then after some time the young one is born, as we get in case of human being also. It is called viviparous condition. After that, of course, both male and female individuals are providing 50% of the genetic material. But you see, a female gamete has to travel little distance, but a male gamete has to travel a larger distance. So nature has developed a very interesting thing, that during gamete formation, in male gamete formation, first meiotic division takes place, then second meiotic division, 4 cells are formed. After that, a cytoplasmic change takes place, in which these spherical cells are changed into elongated cells, sperms. The process is called spermiogenesis. Here there is a negligible cytoplasm in the negligible yolk in the sperm cell. But in case of female gamete, what happens? There is a need to retain much more amount of reserve food material for the developing embryo. So what happens here? Of course, there is meiosis, first meiotic division and second meiotic division, but the yolk is protected, saved. So what happens? In the first meiotic division, 2 cells are formed, a polar body which does not contain any yolk, and all yolk is retained in this secondary oocyte. And then in the second meiotic division, once again meiosis takes place, another polar body is formed, but all yolk is retained, which is utilized by developing embryo in the future. Now, let us see what are the function, responsibilities and challenges to be faced, and accordingly how the reproductive organs are made on the same pattern. One very interesting thing is that in all our exposed areas, for example in the respiratory system, there is possibility of entry of foreign organisms. So here mucus is there, a slightly acidic environment is kept. In case of our buccal cavity, mouth cavity also, saliva is slightly acidic, which kills any incoming foreign microorganism. And even in the stomach also, the digestion takes place in acidic medium. After that, it takes place in alkaline medium. In reproductive organs also, both the sperm duct and oviduct, they are exposed to external environment. They are exposed to entry of foreign microorganisms. So what should be there? An acidic medium is kept in both oviduct and sperm duct. But that acidic medium can kill sperm and ova. So some system should be there that before acidic medium destroys the sperm and or egg, it can fuse with each other to form a zygote, and then further development takes place. So accordingly, the reproductive organs are constructed on this plan. For example, in female reproductive system, there is one pair of ovary and there is oviduct. This oviduct is differentiated into 3 parts: a vagina, a uterus, and this part is called fallopian tube on the name of its discoverer, Fallopius. In ovary also, development of ovum has to perform two responsibilities. One, after meiosis, it has to produce a haploid egg, and along with that, it has to accumulate much more food, reserve food yolk for development of future embryo. So here what happens? At the time of a birth, a girl child contains about 5 lakh cells called primordial follicles, which can give rise in future an egg. But out of these 5 lakhs primordial follicles, only about 500 cells reach up to final stage of development. Rest of them die in between. Developing follicle draws nourishment from surrounding cells and it enlarges. After metabolism of the cells, a fluid-filled cavity is formed, antrum. It is called mature Graafian follicle. Then what happens? This mature Graafian follicle comes near the surface of ovary and membrane of ovary ruptures. It is released. It is called ovulation. Then the remaining cells of Graafian follicle form corpus luteum, which secretes progesterone hormone for further maintenance of pregnancy. In case of male reproductive system, what happens? Here also we have a pair of testes. Development of sperm needs a lower temperature. So what happens? In mammals, the testes just come out of the rest of the body cavity in a small cavity called scrotum. Here the temperature is kept less than abdominal temperature, maybe sometimes 7 degrees less than. And as I have already said you see developing sperms are formed in this testis. We have many coil tube-like structures in this testis, and when we cut a transverse section, we have the seminiferous tubules containing two types of cells. One the Sertoli cells and another the germ cells, which after meiosis give rise to sperm cells. Then in between seminiferous tubules we have some Leydig cells or interstitial cells which secrete male sex hormone testosterone. Now what happens? Here, just after emergence from the seminiferous tubule, the sperm is not capable of fertilizing the ovum. Some changes take place during its journey, which is called capacitation of sperm. It travels from the small coil tube-like structure epididymis, then in vas deferens, and this vas deferens again comes at this stage near urinary bladder. Urinary bladder also releases a small duct which combines with this sperm duct. At this stage, some accessory male sex organs are there: seminal vesicle, Cowper's gland or bulbourethral glands, prostate glands, which secrete certain chemicals which provide nourishment to the developing sperm. And you can see that both duct coming from bladder and the sperm duct unite to form the urethra. So urethra is the common passage for passage of both sperm and urine. Now what happens? This sperm has to reach up to middle part of the fallopian tube. Ovary releases ovum. It travels up to this middle part. It does not proceed further because the acidic environment in this part may kill the ovum. So the ovum is retained here. The sperm has to reach as much close to the ovum, so little contact of acidic environment should be there. It also should not die. For that system, in case of mammals, we have this structure which is called penis, which helps, it penetrates into the vagina and then transfers the sperm here. Sperm reaches up to the middle part of the oviduct and then zygote formation takes place. After that, it starts dividing, 1 cell, 2 cell, 3 cell, 4 cell. This zygote contains only that much reserve food material which is sufficient for development up to 8-9 days. So on 8th day, when it is of the blastula stage, it reaches here. One interesting fact is there, that of course the developing embryo is half genetic material of the mother and half genetic material of the father. So it is a foreign material for the mother uterus. And you know, if my finger is cut, your finger is cut and try to be attached to my finger, it will not work. Our body will reject it. So here also developing embryo is a foreign object for mother. So for 8 days, progesterone hormone secreted by the corpus luteum prepares the uterine epithelium for attachment, and it removes the hurdle due to foreign material of the developing embryo. It prepares the uterine epithelium for attachment of the blastula stage embryo, which is called implantation. Then after implantation, placenta is formed and it starts developing further up to 9 months. But suppose if in a human female, out of 28 days, only one mature ovum is released. In 1 month, one ovum is released from this ovary is kept silent. In the other month, this ovary releases a ovum, it comes into the oviduct. And the life duration of a sperm and egg, both is only up to 24 hours. So what will happen? If in 28 days only one ovum is released, if a sperm meets it, forms a zygote, then a blastula is formed, it attaches with the uterine wall. But if there is no sperm, then the preparation of uterine epithelium by the progesterone hormone for attachment of the embryo is useless because there is no embryo. So what happens? This uterine epithelium, just the progesterone secretion is switched off for some time, decreased, and then what will happen? It will fail to support the uterine epithelium, so uterine epithelium will be released bit by bit and it causes the menstrual flow for 5 days. So this much about it. Thank you very much.